Dear viewers, welcome to online courses. In this segment, we will discuss Xanthophyta. Xanthophyta is a group in algae. It is a division having orange-yellow color. It is a group of algae. As a rule, it possesses cells but not tissues, organs and organ systems. The individuals included in this group have cellular level of organization. Let us see distinctive features of Xanthophyta. First distinctive feature is its habitat. All members of this group are aquatic and are seen commonly in freshwater habitats, and most of them are freely floating. Few members are found to grow on mud and also on the walls of tree trunks. Some members grow on the moist soil and few are marine. Second important character, the plant body is unicellular or multicellular. Multicellular bodies are also organized in different ways. Some are palmelloid, dendroid, cocoid or rhizoidal. Some are filamentous and some siphonaceous. Third important feature is about their cell wall. Their cell wall is often absent, but when present, it contains pectic compounds. Some members have cellulose. In some non-motile forms, cell wall is silkified and made of two halves which overlap each other. The motile forms bear usually two flagella, but rarely one. The flagella are unequal and inserted at anterior end. The flagella are of two types. One flagellum is very large and is whip-like, and the other is short and has lateral outgrowths, having hair-like appendages. Another important feature is their cells. The cells are eukaryotic, having plasma membrane, compartmentalized protoplasm and well-organized endomembrane system. Endomembrane system includes endoplasmic reticulum, dictyosomes, lysosomes, microbodies and other organelles. The chromatophores are discoid in shape and are numerous. The paranoids are absent or rarely present. Plastids are yellow-green in color. The photosynthetic pigments include chlorophyll A and chlorophyll E. In addition to chlorophyll, there is carotene and xanthophylls. The chief xanthophyle present in diadeinoxanthin. The other xanthophylls are violoxanthin, lutein, neoxanthin, flavoxanthin and flavicin. The carotenoids are normally present in excess amount than chlorophyll, which gives them yellow or orange color. Chlorophyll B is absent. The reserve food is oil, lipid or leucosin. Starch is not formed. Plants reproduce commonly by vegetative and asexual means. Vegetative reproduction takes place by cell division, and cell division is always mitotic, involving formation of spindle fibers and organizing in karyokinesis followed by cytokinesis. Karyokinesis as a rule involves prophase, metaphase, anaphase and telophase, whereas cytokinesis is centrifugal. Asexual reproduction occurs by zoospores, aplonospores or akinets. Zoospores are motile spores having flagella. They are thin-walled and generally colorless. Aplonospores are spherical spores having a very thin cell wall. They are produced during slightly unfavorable conditions. Akinets are thick-walled, unicellular, structures produced during unfavorable conditions. They have high power of resisting extreme changes in environment. Sexual reproduction, though rare, may be isogamous, anisogamous, or oogamous. Sexual reproduction occurs with the help of specialized sex organs. Isogamy and anisogamy are found in Botrydium. The Dugin Xanthophyta includes more than 600 species. Xanthophyta completely lack brown pigment called fucoxanthin. Like other chromists, they lack chlorophyll B. This gives them a characteristic yellow-green color as opposed to the golden color of their relatives, which may make them difficult to recognize as chromists. Many xanthophytes produce a cell wall, though it is not composed of cellulose as in plants or chitin as in fungi. Let us now discuss one representative example, that is Vaucheria. So far as systematic position of Vaucheria is concerned, it belongs to domain Vaucheria, kingdom Plantae, Dugin Xanthophyta, class Xanthophyceae, order Vaucheriales, family Vaucheriaceae, genus Vaucheria, and species many. So far as occurrence of Vaucheria is concerned, Vaucheria is cenostic Xanthophycean alga seen usually in freshwater bodies. The alga has 104 species, most of which are seen in freshwater habitats. Some species are terrestrial and few are marine. The terrestrial species like Vaucheria salicylis, Vaucheria hamata, and Vaucheria terrestris form green mats on the moist and aerated soil. Some species grow on exposed clayey soils, while others grow under amphibious conditions. Vaucheria genosi is found on the snow-covered surfaces. About 25 species of this genus Vaucheria are seen in India, which include Vaucheria amphibia, Vaucheria polysperma, Vaucheria unicinata, Vaucheria geminata, and Vaucheria sessilis. So far as thallus structure is concerned, Vaucheria is yellowish-green filamentous alga having aseptate and multinucleated, that is cenostic thallus with profuse branches. It is similar to fungi. The filaments show apical growth and attain a length of several centimeters. Every filament is aseptate, but septate or cross walls are formed at the time of formation of reproductive structures or at the site of injury. However, in some species, the filaments are septate, having cross walls, and therefore cells are present. Terrestrial species remain attached to the soil surface with the help of branched processes called hepterons or rhizoids. In some species, the thallus is differentiated into a well-developed underground system consisting of rhizoids and the aerial part. The wall of the filament is relatively thin and lacks elasticity. It is differentiated into an outer pectic and an inner cellulosic layer. Cell wall is followed by plasma membrane that encloses well compartmentalized protoplasm. That is, inside cytoplasm there is endomembrane system organized in the form of membrane bound organelles like endoplasmic reticulum, Golgi bodies called dictyosomes, lysosomes, mitochondria, plastids, and nuclei. There is a well developed siphon like central vacuole within the filament. It runs throughout the length of the thallus except near tips. The vacuole is filled with vacuolar sap that contains lipids, degenerated chloroplasts and crystals. The protoplast is present in the form of continuous layer between cell wall and central vacuole and is distinguished into outer colored part and inner colorless part. The outer part contains large number of small yellow-green, discoid, oval and elliptical plastids called xanthoplasts or chromatophores. They contain chlorophyll A and E, carotenes and xanthophylls like saphionin and other xanthophylls. The color of the thallus is due to xanthophylls. The inner part of the protoplasm is colorless and contains numerous spherical nuclei, mitochondria, and in addition there is endoplasmic reticulum and other organelles. Chromoplasm is colored, centroplasm is colorless. Chromoplasm plays a role in photosynthesis. Centroplasm is the governor site where nucleus is situated, where from information flows to all other parts of the protoplast. So far as reproduction is concerned, Vaucheria reproduces by vegetative, asexual and sexual methods. Vegetative reproduction occurs during highly favorable conditions and involves formation of septae followed by breakage of filaments into small fragments. This process is called fragmentation. Fragmentation can occur either naturally or through some mechanical agencies. For example, certain insects present in the lakes, ponds, etc. They eat up the alga, leaving behind some parts of the filaments. The parts of the filaments grow into new filaments and give rise to new individual. Sometimes it has been observed that torrential rains also break the filaments. Once the filaments are broken, they grow into new individuals. But this is possible only when conditions are highly favorable. So far as asexual reproduction is concerned, Vaucheria reproduces asexually by forming different spores depending upon the environmental conditions. The spores include zoospores, aplanospores, akinets. Zoospore formation, when does it occur? It is seen in aquatic species and occurs during favorable conditions. In this type of reproduction, the tips of vertical branches swell up and then form a septum. The septum separates a part of nucleated protoplast from the thallus. It becomes club-shaped and organizes itself like a reproductive body called sporangium. Its central vacuole gradually decreases in size so that the nuclei gradually move towards the center. It is followed by reappearance of central vacuole that pushes the nuclei towards peripheral position. The chromatophores move from the periphery towards the center. The sporangial protoplast now shrinks slightly and produces numerous flagella. This multiflagellated and multinucleated body thus formed is called zoospore. What you remember here is that zoospores are multinucleated and multiflagellated, and therefore they are better called synzospores. It has no eye spot, no paranoids. At the apex of zoosporangium, the wall gelatinizes, forming a narrow aperture. Zoospore escapes through this pore early during morning hours. Now let us see the features of zoospores. Zoospores produced by Vaucheria are large, ovoid or spherical, multiflagellated and multinucleated reproductive bodies, lacking eye spot and paranoids. Therefore, they are more accurately called synzoospores or compound zoospores. They possess a central vacuole that remains surrounded by cytoplasm containing inner zone of chromatophores and outer zone of nuclei, reverse of what is present in the thallus. Opposite to each nucleus lie on the surface two flagella of unequal length. Now let us try to understand germination of zoospores. The zoospores produced by Vaucheria swim for about 15 minutes after their release from the zoosporangium and then come to rest, withdraw flagella and secrete a thin wall around them. Therefore, they become deflagellated, non-motile and sluggish. Soon they produce 1 to 2 tuberous siphonaceous outgrowths. Instead of mitosis, there is endomitosis, that is karyokinesis takes place. It is not followed by cytokinesis. That's what leads to formation of multinucleated condition. In terrestrial species, one of the outgrowths attaches itself to the substratum and functions as holdfast, whereas the other forms erect branched part. Now let us try to understand aplonospore formation. As I have already talked, aplonospores are non-motile spores produced during slightly unfavorable conditions. They are commonly formed in terrestrial species of Vaucheria, but in aquatic species, such spores are produced under unfavorable conditions. Like zoospores, aplonospores are formed singly within aplonosporangium. Aplonosporangium develops as a club-shaped structure at the tip of a young vegetative branch. A transverse septum cuts off the terminal sporangium from the rest of the filament. The protoplast of the sporangium metamorphoses into a single multinucleated aplonospore. Aplonospores are produced by other algae also, but the difference here is that the aplonospores of Vaucheria are multinucleated. The aplonospore is a thin-walled, club-shaped or globose structure. It is liberated by rupturing the apical wall of the sporangium. Sometimes the protoplast of the aplonosporangium divides into a number of spores. These small, thin-walled aplonospores are called microaplonospores. The aplonospore or microaplonospores germinate to form a new thallus. Again here, I will mention that it is endomitosis that favors their growth. Another type of asexual reproduction is formation of achaenids. Some terrestrial and aquatic species which are subjected to unfavorable conditions, the contents of their filaments divide into small segments by thick gelatinous walls. These thick-walled multinucleated segments are called achaenids. So, they are not necessarily spherical or oblong, but they can be filamentous. They are also called cysts or hypnospores. Their protoplast is often laden with oil droplets. They germinate under favorable conditions and give rise to new thalli. Here I will tell you that achaenids are highly resistant. Even if a water body dries up, they have ability to survive. They have ability to be carried by running water. Sometimes the protoplast of these acanites may divide into many uninucleated amoeboid segments. These segments come out through a pore in the lateral wall of the cyst. At this stage, Vaucheria thallus resembles to that of another genus called Gongrosera, and therefore this stage is called as Gongrosera stage. So far as sexual reproduction is concerned, Vaucheria reproduces sexually, which occurs during unfavorable conditions. It always involves oogamy and takes place with the help of specialized reproductive bodies. Male reproductive bodies are called antheridia and the female reproductive bodies are called oogonia. Most of the species are homothallic or monoecious, but some are heterothallic or dioecious. The reproductive bodies usually develop on filaments growing on damp soil or quiet water. Stagnant nature of the water is important. In homothallic species, antheridia and oogonia are born adjacent to one another on common lateral branch or adjacent lateral branch. They are stalked or sessile. Some species show variation in their relative position. For example, Vaucheria geminata, you will see in it central stalked antheridium surrounded by 3 or 4 oogonia. In Vaucheria gardneri, each antheridium has many oogonia at its base. Now let us try to understand what is antheridium. Antheridia are male reproductive bodies. They develop on main filament or its lateral branch as strongly curved cylindrical structures. Large number of chromatophores and nuclei accumulate in this region, and this curved part is then cut off from the rest of the filament by a septum. The nuclei present in this region divide endomitotically, and then the protoplast divides to form uninucleated segments. Each segment metamorphoses into a spindle-shaped biflagellate antherozoid. The two flagella are of unequal length, dissimilar, and laterally inserted. The antherozoids do not have chloroplast and also lack eye spot. They are liberated through an aperture at the anterior end of the antheridium. In Vaucheria debariana, there are usually several pores on lateral wall of the antheridium through which antherozoids liberate. Now let us try to understand oogonium and its development. The oogonium is the female reproductive body which initiates as a small protuberance at the base of the antheridium. Suppose this is the position of antheridium. At the base of antheridium, oogonium is formed as a protuberance. It arises due to accumulation of cytoplasm and a large number of nuclei. The dense multinucleated mass of cytoplasm thus formed is called wanderplasm. It gradually increases in size and forms an ovoid or a rounded oogonium. The oogonium is separated from the subtending filament by a septum. The mature oogonium is spherical or subspherical with an apical beak. It has a single nucleus, the other nuclei present in the young state degenerate. Degeneration occurs after septum is laid down. However, some workers believe that all nuclei except one pass out of the oogonium before the formation of the septum. The uninucleated protoplast of a mature oogonium forms an egg cell. At the anterior end of the egg, there is a hyaline area which acts as a receptive support. Now, let us see how fertilization takes place. As a rule, fertilization is favored by water and occurs in water. During fertilization, antheridia and oogonia mature almost simultaneously. The tip of the apical beak of oogonium gelatinizes to form an aperture. Simultaneously, a large number of antherozoids are liberated from the adjacent antheridium. A drop of colorless cytoplasm comes out of the beak of the egg, and a number of antherozoids get entangled in the drop. Only one of the many antherozoids enters the oogonium and finds its way into ooplasm through a receptive spot. The nucleus of antherozoid is smaller than the nucleus of the egg cell. After entering the egg, the male nucleus gradually increases in size and becomes nearly equal to the size of egg nucleus. Then the two nuclei fuse to form a diploid zygote. The zygote secretes 3 to 7 layer thick wall. You must remember here that the conditions are unfavorable, and the alga has ability to pass through unfavorable conditions through the formation of either akinets or zygospore. Even if pond dries or a water body dries up or the temperature conditions change, zygospore or the akinets, they remain persistent. The protoplast of the zygospore is laden with oil droplets. It is green in color initially, but gradually it turns red due to degeneration of chlorophyll. It remains in the oogonium until it is liberated by the decay of oogonial wall. The zygotes remain dormant for a few months before germination. The dormant state helps the alga in tiding over unfavorable conditions like desiccation and high temperature. Now, let's try to understand how a zygote or zygospore germinates. Once unfavorable conditions are over, dormant period is over, zygospore germinates to form new thallus. First of all, before the germination starts, it is thick cell wall having many layers start degrading and loosening. Then the diploid nucleus divides by zygotic meiosis to form 4 haploid nuclei. It is followed by repeated endomitotic divisions, which results in the formation of a synostic thallus. Here I would like to mention, one nucleus starts endomitosis, produces many nuclei, but simultaneously it is followed by prolongation of the protuberances and elaboration of the thallus. Once thallus is formed, reproductive bodies are again established and the cycle is repeated. From this, we conclude that the life cycle of Vaucheria is purely haplontic, characterized by zygotic meiosis and all other stages haploid. Gametes are formed mitotically and are therefore mitogametes. This is all about Vaucheria. Hope you have enjoyed the lecture and have understood the basic concepts. Thank you.